Hey everybody, Happy New Year. Let's talk about the immune system. All right, these are all the things that we're going to be talking about in this particular order. All right, let's just start with a general overview of innate versus adaptive. There's no perfect place to start, so at some point throughout this lecture, we're going to cover ground that we've already tread, but I'm going to try to slowly introduce a lot of the ideas to you. So some of this we should be familiar as well with, from the MCAT and undergrad. The innate immunity is going to be something that's just normally present. You don't have to activate it. It is not specific. Because it's not as specific, it has a lower potency. It's not as effective, but it's very rapid. It has no memory, and allergic reactions have nothing to do with the innate. On the other hand, the adaptive is going to be specifically catered towards one particular organism, one particular pathogen. So it's specific. That makes it a high potency, but it takes a little bit longer to develop. Hypersensitivities and allergies are going to be adaptive, and there is a memory for the adaptive, not the innate. You can think of the immune system in general as starting with these physical barriers that pathogens have to permeate, and then once they get through they break the wall down, then they've got to deal with the police officers or whatever who are patrolling, who are on duty, the macrophages and the neutrophils, etc. And then, at the end of the day, or the last line of defense is going to be the adaptive immune system, the T cells, the B cells, and whatnot. So within the innate immunity, our major players, we have our granulocytes, mast cells. I would also think, though, the mast cells, basophils, and eosinophils, you can kind of group them together because they're going to be doing, neutrophils degranulate as well, but I would say that mast, basophils, and eosinophils are degranulating a bit more. Natural killer cells technically are a lymphocytic origin, and yet we consider them part of the innate immune system, so that's unique. The granulocytes typically are going to be, bilobe nucleus or a polylobe nuclei. The macrophages and dendritic cells, which come from monocytes, are going to be mononuclear. Complement proteins are also going to be innate. A lot of this stuff, though, there is going to be a connection. Like the complement system can be exacerbated and amplified by the adaptive immune system. And then, we have our bread and butter, our B cells and our T cells, which can be broken down into CD4 positive and CD8 positive. B cells versus T cells. B cells are going to be the humoral response, which for the most part means that they're floating around in the blood. So typically we're going to have some sort of antigen being presented by a macrophage that activates a CD4 helper cell. The helper cell then is really a major, it's like the quarterback of the immune system. It can start, further activating, empowering B cells, activating CD8 cytotoxic cells. And the reason I have this picture, I think of the Nazis, the Gestapo going door to door. That's sort of what cytotoxic cells are doing. Is they're going door to door, cell to cell, saying, who's in here? Is there a virus in here? Is there a cancer in here behind this door? And for that reason, the T cells are more viruses, cancers, and the B cells are going to be outside the cells, bacteria. Now, there could be an exception to that if we're talking about something like a tuberculosis, or mycobacterial pathogen, something like, or listeria as an example, could be part of the cell-mediated T response. So this is really just showing the same thing. T cells are going, personal, checking who's inside, knock, 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 and if you're somebody we don't want, then they're going to kill them. Antibodies really can't get inside of the cells, which is why they're the humoral. They're going to be floating around in the blood and the lymph and maybe the interstitial fluid. So every B cell and T cell, as we're probably very well familiar with, has a soulmate. This antibody is specific for this, not 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 for any of the other, millions of possible antigens. It's just going to be for the A. So that means that we have a one antibody that's specific for tuberculosis, another one for Zika, another for some Mars virus, which, doesn't even exist yet. We haven't even encountered. It's there. It's waiting. And what applies for B cells is also going to apply for the T cells as well. The main reason for this is because we have VDJ recombination, which we'll talk about soon, later. All right, this is a kind of cool picture of phagocytosis. He's got him right there. Oh, that's badass. Point being with this is that macrophages, they don't have to wait for permission. They see a bad guy, they can just go in and they can attack. Can T cells do that? No, T cells have to wait for something to be presented on a silver platter, or more specifically, an MHC. So CD8 is going to respond to MHC1. I think of 1 times 8 is 8. On the other hand, MHC2s will respond for CD4 cells, and I think of 2 times 4 is 8. The reason for this, the reason that a T cell can't just go up to a pathogen, to an antigen, and attack it is, it's we're trying to cut down on autoimmune diseases. So it's by having the macrophage there, it's kind of saying, like, are you 100% sure you want to be activated by this antigen? Because that means forever. Now we're going to have memory cells. And if you're getting, mistakenly activated by collagen, then that's going to be super bad. So it'd be like if you were drunk and you were going to do something silly, and then you had to have your friend check you and say, hey, I've got to give you permission to do this. All right, continuing the overview kind of, and then we're going to get into a lot of the newer specifics that people, that's the new material. So all of our immune cells are going to be made in the bone marrow by hematopoietic stem cells. They have 2 major branches that they can differentiate into, the myeloid and the lymphoid. The lymphocytes will be the T lymphocytes and the B lymphocytes. Also, though, the natural killer cells. Natural killer cells, again, are innate, but they come from the lymphocyte progenitors. The myeloid is going to be our, megakaryocytes, make platelets. And then we have our granulocytes here, which are neutrophils, eosinophils, basophils. And then the monocytes can become the dendritic cells and the macrophages. All right, bunch of proteins we need to know. T cells, of course, have a T cell receptor. Whether you're a CD4 or CD8, you always have a CD3. So CD28 is going to be really important for activation of CD4 cells and to some extent CD8 cells. They're going to interact with the B7, which is sometimes called CD80 or CD86, on an antigen-presenting cell. And so this is just this is called co-stimulation, and this is just another way to as much as possible avoid autoimmune diseases by requiring this second receptor. If they're not co-stimulated, something called anergy happens, which is basically where the B or T cell is deactivated. Helper cells, CD4, because they have the cluster of differentiation is the name of the protein that's on the cell membrane. They have the CD40 ligand. And then this was the thing that was, I think in China a couple of years ago, they did the first babies were CRISPRed, and they were specifically had these genes cut out, which made them slightly more HIV resistant, which is obviously a big ball of ethical worms. Cytotoxic CD8, regulatory T cells. These aren't really that important, but they come from a helper progenitor, so they're also going to be CD4. B cells, anything around 20. So 19, 20, 21, that's going to be a B cell. B cells are also antigen-presenting cells, which is kind of weird. Macrophages, B cells, dendritic cells are going to be antigen-presenting, which means they have an MHC2. They have a B7. Remember that B7 is what activates our T cells. B7 is sometimes called CD80. One way that I think about this is that because they have an MHC2 times 40 is 80, and these are all things that we will find on antigen-presenting cells. CD14, specifically going to recognize lipopolysaccharides. This is a part of sometimes these are called TLRs, toll-like receptor. Again, these are antigen-presenting, so they have a 40. And they can bind to FC, which is, if I'm looking at an antibody, this is my FC, the constant region, and then they complement 3B protein. So all of these are going to be opsonins. There are 2 opsonins, the IgG FC and then the C3b, which just makes phagocytosis a little bit easier. Natural killer cells, they're the only CDs that I can think of that have a 6 in them. And so I always thought of like a 6-shooter gun. And that's natural killer cells go around and they kill people. That's what they do. Hematopoietic stem cells, I always thought of like C, D, that's the 3rd and the 4th letter of the alphabet. And that works pretty perfect. And who's perfect? Babies are perfect. Babies are perfect, and then people grow up and they get a little shitty. So hematopoietic stem cells are going to be the baby cells, and that's how I remember that CD34 is the marker for them. Here's just a picture of our CD28 with the B7, also known as the 80, and again, 2 and 80, and then we also have 40. And then the role of the 28 and the 7 is going to be activate. The role of the 40 is the T cell is empowering a cell. So this is going to be a CD4 cell, and it's sort of increasing the power of this B cell, which we'll talk more about later. General picture of what's happening. So a pathogen gets through our first layer. It gets through all of the lysozyme sweat on our skin. We have bacteria in our skin which are competing with them, like good bacteria. They have to penetrate the skin itself. So they get through all of those first layers, and now they come in here and they've got to deal with the macrophages. Macrophages are going to be our police officers. They are, the first line of defense. They're sitting there, they're waiting, and a battle ensues. The macrophages release cytokines. These call for reinforcements. So neutrophils are always going to come in second. Neutrophils will start to migrate in from the blood. They will enter into the battle and start fighting the bacteria as well. Meanwhile, a dendritic cell, which is very like a macrophage, will leave the battle, but it has samples of the pathogens. It's holding a bunch of samples, and it's going to present them on its MHC2. It moves over to a lymph node. So it moves to a lymph node where it has helper T cells and B cells and things like that. It finds the perfect helper T cell that recognizes its soulmate. Maybe this is like a tuberculosis infection. It activates it. That helper T cell can then help stimulate B cells to become plasma cells. A plasma cell is just an activated B cell. That releases antibodies. We remember that is the humoral response. At the same time, the helper T cell might activate a CD8 cytotoxic cell, which is going to go ahead and kill the virus or the tuberculosis or whatever it happens to be. Also, when we're initiating the inflammation, we're increasing the diameter of our capillaries, so more blood is going to flow in. We start to make it a little bit leakier, so this blood can actually leak through, and with it complement proteins and antibodies, and as we see here, neutrophils can enter into the battle. So antigen presenting cell, again, that's a macrophage, a B cell, or dendritic cell, encounters a pathogen, an antigen, it phagocytosis, and then it presents it on its MHC2. That activates a helper T cell. Helper T cell can then activate B cells. They can also activate CD8 cells. The CD8 cells can then go ahead and kill people. CD8 cells work very similar to natural killer cells, so they're just going to go ahead door to door like the Gestapo and just kill whoever's inside. The B cells become plasma cells, also become memory cells. And the memory is really important because if we encounter that same pathogen again, we're prepared. We're much better prepared than the first time, which is often why they say you cannot catch the same, disease twice. Often you really can catch it twice, but it's such a mild infection the second time that you never really know it. And again, T cell mediated, more likely to be viruses, cancer. B cells, humoral, more likely to be bacteria floating around in the blood. Again, we're looking at our initial response. Initially, we might have there's a soulmate. We might have a B and T cells specific for tuberculosis in our body somewhere, all of us, whether or not we've been infected, but we just have a few of them. And so when you get infected, it takes a while to find those cells, and then it takes a while to amplify them. So, it might take 7 whole days before we can actually start getting some, antibody production and some reasonable immune defense mounted. That's also the IgM. So IgM is going to be the primary response. But now we have memory cells. So if I encounter the antigen a second time, I already have kind of like a standing militia that's ready to target this specific pathogen, these memory cells. And so the response is going to be much quicker and much larger, which is again why you don't really notice the disease the second time around. And these are going to be IgG the second time. All right, myeloid progenitor. I think it's a good idea to know what a lot of these things look like. We'll talk about this a bit, but of course, erythrocytes are part of the myeloid system, but they have, we see, evacuated their nucleus. If you see a Heinz body or some erythrocyte with a nuclear remnants, that means your spleen's typically dysfunctional. I would also note the neutrophil as this N-band. The N-band is going to be an immature neutrophil, and if I see a lot of N-bands, that means that's there's likely an infection somewhere in my body, and my stem cells in my bone marrow are working too hard to produce all of these leukocytes, these white blood cells. All right, monocytes. We said dendritic cells and macrophages, also osteoclasts, which kind of makes sense. Macrophages eat pathogens, and osteoclasts sort of like eat bone. So. what do macrophages do? Macrophages phagocytize. That's what, macrophage, big eater. They're going to, engulf pathogens like so. They're going to incorporate them. They'll bring them into vesicles. They're then going to digest them. Usually they'll fuse it with a lysosome, so a phagolysosome. We break it up. We take, these little tiny antigen remnants, and then we put them up on this silver platter for the T cells, MHC2. At the same time, we're releasing a lot of cytokines signaling, trying to increase inflammation, trying to recruit other cells, IL-1, IL-6, TNF-alpha. We'll talk more about these later. Also, to some smaller extent, they produce IL-8, IL-12. There's a couple different names for macrophages. In the liver, sometimes we call them Kupffer cells, Langerhans cells. Some people call these macrophages. I think they're more like dendritic cells, osteoclasts in the bone, and microglial cells in the brain. Again, all of these are basically macrophages, except this one, which is more of a dendritic cell. And CD14, we say, can bind to a lipopolysaccharide, which is something that bacteria have on their outside. And if this happens, we'll have a systemic immune response, which is actually not a good thing, because part of the immune response is usually vasodilation. But if it's bacteria in your blood, that means it's systemic. It's everywhere. What's Boyle's law? If the volume of my blood vessels increases, then the pressure goes down. So this super low blood pressure is going to cause septic shock. Septic meaning like from bacteria. And so this hypotension can actually kill you. They're showing the receptors here. Sometimes we say that they're toll-like receptors, the things that they're interacting with. The things are PAMP, pathogen-associated molecular pattern, something that would only be in a bacteria. Flagellin is, we have tubulin in our microtubulus, they have flagellin in theirs, and peptidoglycan is surely not going to be on our eukaryotic cells. Dendritic cells are the link between, so they're technically part of the innate immune system, but they're going to go to the lymph nodes and activate our B cells and T cells. So this picture is a little bit convoluted, but just look at this right here. So if I have an infection, I have inflamed skin, the dendritic cells are going to pick up some of the remnants, some fallen warrior's head or arm or whatever. They're going to present it on their MHC2, and they're going to travel to the lymph node. And here we have these B follicles. When they're like this, they're inactive. They don't have germinal centers. But now that the dendritic cell comes in, we're going to go ahead and activate these puppies. They're going to get enlarged, the lymph nodes get swollen, and then we're going to send, activated B cells and activated T cells out to help fight the battle. All right, we've already talked about this a lot, but I really want to hammer it in. CD28 is to activate a T cell. That's why this one is naive. It pairs with CD80, sometimes called 86, sometimes called B7. We know that the CD4 goes with the MHC2. Why? Because 2 times 4 is 8. And then the CD8 goes with MHC1. Why? Because 8 times 1 is 8. I like to remember that there are 3 antigen-presenting cells: dendritic cells, macrophages, and B cells. Again, it's weird that B cells are antigen-presenting, but they are. 40 times 2 is 80 because they have a CD40, a CD80, and an MHC2. Again, 28 is the activation. And 40 is once you have a CD4 that's been activated, it can power up other cells. So power up, let's talk about this. There are new cancer therapy drugs out there, which are really just agonists of CD40. Well, how would that help or work? Again, if CD40 is activating, it's powering up the immune system. We're powering up our macrophages and our neutrophils and our natural killer cells and our dendritic cells and our B cells, just supercharging them so that they can kill the cancer. Now, you don't really have to know this specific example for your NBME coming up, but I think just very perfectly illustrates the CD40 power up thing. And I just think of Mario, where you can get all of these different power up things. So MHC1 versus 2. So we'll see a picture of this in a second. So I'm going to ignore the structure here. MHC is present in all cells. Well, MHC2 is only, again, on antigen-presenting cells. We said macrophages, B cells, and dendritic cells. MHC1 is taking endogenous antigens. So just the things that a cell is normally transcribing and translating, that's what it puts on its MHC1. It's giving kind of a snapshot to the interior of the cell. The MHC2 is like, I killed a man and then dismembered his body, and then I'm presenting his arms and his feet and his head or whatever. That's what the more barbaric antigen presenting cells are doing. MHC1 to CD8, 1 times 8 is 8, and CD4 is going to the MHC2. Talked about that. And Yeah, cool. Next one. Sorry, here's a picture. This is MHC1. I actually would know what this looks like. So we just have this little tiny beta 2 microglobulin in MHC1. MHC2 is just two even Steven, two beta chains and two alpha chains. And again, here I have, we can imagine an infected cell. So how is this even working? An infected cell, let's say it's infected with a virus. It's now producing a bunch of viral proteins. And again, the point of the MHC1 is whatever's going on being produced normally. So even an uninfected cell still has an MHC1. It's still producing antigens. But the idea is there's nothing interesting. It's like producing an antigen that has a polymerase or an actin. And those are normal things. So the cytotoxic cell might come over, again, like the Gestapo, and inspect and say, okay, everything checks out here. This is just an actin. This is just a polymerase. But if I have an infection, well, what is my antigen going to be now? It's going to be some sort of viral protein, and that signifies the cytotoxic cell. Everything is not fine, and then it should kill that cell. Note that the exact same thing would happen if we had a cancerous cell. So it's not just is it a normal antigen or an abnormal antigen. It might be, hey, there's way too much, let's say, there's too many nuclear proteins, and that might be a signal that there's too much replication happening because this is a cancer cell. And so that's another way that a cytotoxic cell can kill this, because it's not infected, but it's cancerous. And by measuring the frequency of an antigen on the MHC1 on the outside, the cytotoxic cell can make that executive decision or not. Again, we already talked about this. This is just activating our helper T cells. All right, so MHCs, whether one or two, they are made up of HLA proteins, and we have a bunch of different subtypes. So we have A, B, C. So A, B, C are going to be our class 1 genes, but then we also have our MHC2 genes, DP, DQ, DR. I just remember this, that A, B, and C are just one letter, and so that's for the type 1 gene, and DR, DQ, and DP are 2 letters, and so those are going to be for the class 2 genes. Again, these are just making up the molecule over here. Just another picture kind of showing the same thing. We have one chromosome and it's got a bunch of our HLA types for one type of MHC and then HLA types for a different type of MHC. So these are going to be highly variable between people, and the highly variable nature is actually good. We want this genetic diversity so that we can have kids who can be perhaps well adjusted to a large variety of pathogens. And this is just something that is very favorable from a natural selection standpoint. And so why do I have this picture of this guy sweating? So it's so important to have a diverse HLA subtypes in your children that they've actually done tons of different studies which show that people are subconsciously attracted to individuals that have a different HLA subtype than you. So if you're HLA A1 and your sister is HLA1 or your brother or whatever, you're less likely to be attracted to your sibling because they have the same HLA type. And again, that's favorable from a natural selection standpoint, because you might, for a lot of reasons, not want to mate with your partner or your sibling, and you want to have a diverse immune system in your offspring. So yeah, you'd rather be attracted to, if you're HLA1, you'd be attracted to someone who's HLA, 2 and HLA 3. And again, this is going to be for, we have A, and then there's going to be a different attraction for B and C. That's not on the test. I just thought that was interesting. Some of these alleles are going to be associated with autoimmune disease. One you should definitely know is HLA B27 is associated with ankylosing spondylitis and a bunch of other of the seronegative spondyloarthritis conditions. Also, the HLA DR1 and DR4 are associated with rheumatoid arthritis. And all right, so natural killer cells. One way that they differ from a CD8 cell. We said that a CD8 cell, its job is to inspect the MHC1 and then just get a snapshot of what's the production of proteins like inside of that cell. And that can tell us if it's infected or if it's cancerous or something. You would think, well, okay, then it would be advantageous for a virus or a cancer then to outsmart the immune system by decreasing expression of MHC1. That way, the CD8 cells can't kill them. Well, that's what we have natural killer cells for. This is a natural killer cell, and it's inspecting. It's just testing for the presence of an MHC. And it goes, yep, there's an MHC there, so check, everything is normal. You have your MHC, you're probably not infected. But in a different case, I guess, sorry, this is the natural killer cell, and then this is the normal cell. So check goes here. But in this next case, notice that we're missing our MHC1. So now the natural killer cell over here decides to go ahead and murder this infected cell. Again, it's assuming it's murdered because it's not producing an MHC1, and it's saying, hey, if you've got nothing to hide, you would have your MHC expressed. And it has perforins and granzymes, which is basically poke holes in the cell, and cells, surprisingly, don't do very well with holes poked in them. Another way that they can be activated, so that's not the only way, is if I have a tumor cell and it's been tagged by an antibody, shown here. The natural killer cell can bind to the antibody and then decide to similarly go ahead and poke holes in this tumor cell. Neutrophils are just the most abundant granulocyte. So of all of the granulocytes, basophils, eosinophils, neutrophils are the most important, most common, and it's important to know again what all of these things look like. So this is like a polymorphonuclear nucleus. It's just got many different lobes. And it's high yield to know exactly. We said a macrophage encounters a pathogen, it sends signals, and then the neutrophil has to come in. So how does this happen? Step one, rolling, is we have something called a Sialy-Lewis X acid, and this is going to, so this little green thing is the Sialy-Lewis, is going to, so here's our, we're in the blood vessel right here, and here is the interstitium where the battle is taking place. The endothelial cells will produce the different selectins. Those will bind to the Sialy-Lewis and slow down the neutrophil. Because you got to remember, blood is like a roaring river, and then the cells are just flying past. So this is going to slow down the neutrophils. So that's rolling and slowing down. Okay, well, how do they stop them? Now we have a protein called ICAM on the endothelial cells, and that's going to match with something called an integrin on the neutrophil. That stops it. So the selectins slow it down. Think S for selectin, S for slow. And then the ICAM is going to completely stop it. And then, well, how does it get through? A different molecule called PCAM is going to allow it to migrate into the area. Now, C5a, IL-8, LTB4, and maybe various bacteria, signatures like lipopolysaccharide, LPS and flagella and things like this. There's going to be a chemotactic gradient, and then they're going to slowly, hone in towards the battle. And again, what started a lot of this, the initial call from help came from the macrophage. These are probably two of the most important signaling factors early during an infection. So because neutrophils come in second after the police officers call for their help, the macrophages, I always thought of them as like a SWAT team. They're also a little bit more badass than macrophages. So chemotaxis, so again, I would know these three chemotactic agents. C5 is a complement protein. This is an interleukin, and then this is a leukotriene. Chemotaxis, we have a gradient. So imagine that this is all just LPS. This thing is shedding LPS. And it's sort of doing like a Marco Polo, getting warmer, it's just moving towards the increasing sound, through this concentration gradient to chase it. You can think also it's smelling its way towards the bacteria. And this is just a cool picture or cool GIF of a macrophage or some sort of cell chasing a little bacteria, and I just think that's so badass. All right, neutrophils. Neutrophils have something called a respiratory burst. So they can take oxygen. They can use NADPH oxidase. Super important step to make it into superoxide. Superoxide dismutase will turn it into hydrogen peroxide, shown here. And then hydrogen peroxide, well, that itself is just very damaging. This and this are both super damaging. They can kill bacteria. But they can also go ahead and convert it into bleach, HOCl, which will, again, just here's my bacteria. Not anymore. I've now poked a whole bunch of holes in this bacteria. So the NADPH oxidase can be, there's a couple diseases that might affect it. NADPH oxidase deficiency causes something called chronic granulomatous disease. The hydrogen peroxide also can eventually be with glutathione or catalase. It can be turned into water. So so if somebody does have a disease where they have a defective NADPH oxidase, in that case, they'll go ahead. Bacteria actually sometimes have hydrogen peroxide, H2O2. So, we can't do this first step, but you know what they'll do? They'll just commandeer the bacteria's hydrogen peroxide and use that to, against its own, producer, the bacteria, and make HOCl bleach, and that'll kill the bacteria. However, bacteria sometimes, some bacteria are catalase positive and some are catalase negative. If you're catalase negative, you can kill the bacteria by stealing its H2O2. But if it's catalase positive, well, the bacteria actually was going to neutralize its own hydrogen peroxide and turn it to H2O. So this is not going to be as effective. And this will be important as well in the bacterial unit, which is coming up. Green sputum. The myeloid peroxidase is a blue-green pigment that neutrophils have, which gives this its lovely green color. All right, couple ways that neutrophils can just be badass. Don't forget, they're granulocytes. They have granules all over their body. And these granules, have things like alkaline phosphatase, collagenase, lactoferrin, a bunch of other chemicals that are not fun, and they will just send them out like a little grenade, and it'll kill the bacteria. On the other hand, they could go ahead and just phagocytize it like a macrophage. They put it into a phagolysosome, which is really just an azurophilic granule, is really just a lysosome. So they'll fuse the vesicle with the lysosome, and that'll be a phagolysosome, and that also kills the bacteria, breaks it up into little parts. This one's pretty crazy. They can just take their DNA, and basically it's like ripping out your own guts and then using that as a weapon against somebody. They'll take their DNA and they form these large little DNA nets, and this will catch bacteria in the nets, and then that kills the bacteria. So the neutrophils, again, they're the SWAT team. They come in after, and they're really good at killing things. But there's a lot of collateral damage that neutrophils invoke on the tissue. So I'm reminded of this movie, Team America: World Police, where the America thinks they're the good guys. They go to all of these foreign countries and then they kill terrorists. But everywhere they go, they always end up, they destroy Paris. That's the Eiffel Tower. They go to Egypt and they destroy the pyramids. And so that's what neutrophils do. They go in and they attack the pathogens, but they also harm tissue. And that's going to be important for just the rest of preclinical. That comes up again and again and again. If you have an infection, you need to produce a lot of immune cells really quickly. And sometimes you push cells out before they're even ready in a desperate attempt to have fighters against the bacteria or whatever. And so this is a neutrophil that's mature. But remember, we said that the band cell is the immature neutrophil. So it gets sent out prematurely because we're just turning out so many, I think of like a child soldier, like someone's invading our country and we just have to send 12-year-olds with, AK-47s or whatever to fight the bad guys because we're just desperate. So this is called a left shift. A left shift is where you have band cells, which just is usually a sign of an infection. Granuloma formation. So I think I'm reminded of the Disney movie Hercules. We have these titans. So the titans have been imprisoned, I don't really know where, like somewhere in Hades or whatever, and in the underworld. And they're imprisoned there, and they're bad guys. That's it. And same thing, if there's an infection like tuberculosis that we're having trouble like really killing it, well, if we can't kill it, let's at least imprison it. And so we put it in this granuloma, and we trap, the bacteria, inside of here. Surrounding this is really just a ton of macrophages. So we're surrounded by epithelial cells, which are just macrophages, and Langerhans cells, which are really just multinucleated cells, sometimes called giant cells. Again, those are also just macrophages that have they've divided a lot, but they didn't split their cytoplasm, which is why they're multinucleated. And then they're surrounded at the end by lymphocytes. There's two types of granulomas. The ones that kind of make sense, we have tuberculosis or a fungus that we're just having trouble killing, so we wall it off. These are said to be caseating. There's a central necrosis, and that makes sense because inside of here, there are bad guys, and they're just killing your tissue. Even though they're trapped, they're still causing necrosis. But then you also sometimes form granulomas for stupid reasons because you have an autoimmune disease. Granulomas are still bad, but these are not necrotic, so they're non-caseating. It's important to know a lot of the cytokine, build up to a granuloma. So to activate a T cell, we have an antigen-presenting cell. It presents this. We can convert a CD4 cell into a specific type of CD4 cell, which is called a TH1. Remember that CD4s are helper cells. So what does TH stand for? It stands for T helper. So this is T helper 1. Later we'll talk about T helper 2. T helper 1 secretes INF interferon gamma. This is going to sort of empower, activate macrophages more than they already were. They now are going to secrete even more tumor necrosis factor, TNF. And, all of this stuff together, but especially this last step, helps form a granuloma. Why is this significant? Because it's really, really high yield to know that a lot of medications for autoimmune diseases, anti-TNF alpha or TNF alpha inhibitors, well, if we're stopping the TNF, we might, free a granuloma, right? So we have these imprisoned people, and if you're on something like Humira or TNF inhibitor, it'll free up these pathogens. The pathogen is often, again, tuberculosis. That's a really common one. So before you put somebody on Humira, you have to go ahead and give them a PPD test or quantiferin test, make sure that they do not have tuberculosis, because otherwise you could put them on the drug and then there could be a reactivation of the tuberculosis. Mast cells contain basophilic granules. They're going to be similar to basophils, which we'll see in a second. And they're going to stain blue, basophil B for blue. And if it's a basophil, it loves bases, which means it is an acid. So it has a lot of acidic components inside. They're going to have IgE antibodies on their surface. So it's, here it's going to have a bunch of these IgEs. And the binding of a pathogen, which cross-links, so this is my pathogen here, P for pathogen. If it cross-links, and it connects these two antibodies, that is going to cause a degranulation where we start to release a bunch of histamine, other molecules as well, but especially histamine. This plays a really large role, as many of you are probably aware, in allergies and just a generalized inflammatory response. So we're releasing histamine, heparin, leukotrienes. And think about the effects of all of this. This is going to dilate blood vessels. This is going to cause the endothelial cells to become a little bit leakier, so fluid from the blood can leak into the interstitial fluid. Heparin is an anticoagulant. What is that going to do? Well, that just means we're going to get more blood flow. There's always going to be some amount of background coagulation proteins, but if we have heparin, we're going to have less of that. So more blood flow can come into this because it's less coagulated. And then the leukotrienes are going to help recruit neutrophils and other cells and allow chemotaxis. Remember LTB4. And this is a type 1 hypersensitivity reaction, which we'll talk about later. So this is our normal picture shown here. But now our mast cells are releasing a bunch of histamine. That is going to allow, notice that this is expanded a little bit. So we have vasodilation, more blood is going to come in. And we have, this was a little tighter, but now we're having a bunch of neutrophils leak through. Also complement proteins are going to leak through. Blood is going to, look at all of this blood coming in. That's why when you have an inflammatory response, something gets really red because of all the blood coming in, and that's why it gets swollen too, is the blood just takes up more volume and it pushes the skin. Here's the skin kind of flat, and here's the skin's kind of pushing out a little bit. Again, you should know what mast cells look like. If we have to say there's one difference between a mast cell and a basophil, I would say that it's, I'm sure there's more differences, but at least for the purpose of your NBME, mast cells might be an acute allergic response, while basophils will be more of a chronic allergic response. So pretty much all the exact same stuff here. I'm not even going to repeat it. Basophils are very similar to mast cells. Difference though, remember that these count as granulocytes. Mast cells technically don't, even though they still degranulate. They have a bilobe nucleus, which we'll talk more about. And if mast cells are the acute response, mast cells are probably the more common, more important one, but basophils are going to be a little bit more on the chronic side. So basophils, in contrast with eosinophils, eosinophils stain red. And eosinophil means that they, love eos as acid. So these love acids, which means they must be a base. These are going to play a role in asthma. They're going to play a role in parasites and allergies too, to some extent, but I would say more the asthma. They have a bilobe nucleus, very similar to the basophils, stimulated by IL-5. And they're activated by IgE. That's really important to know, is that eosinophils are activated by IgE. So if you look at this helminth, this large parasite, this worm, it's too large to be engulfed and phagocytized. So the eosinophil shown here will bind to an IgE and just degranulate, and it releases a bunch of little grenade things to try and damage this parasite. And I think it's really high yield to know, they'll mention this in a vignette, if something's multi-lobed, it's a neutrophil. If it's bilobed, it's an eosinophil. This one doesn't really look bilobed, but we still consider that bilobed. But you can mainly tell because here we have the blue color, B for blue, and then we have the red color over here. Monocytes, while we're here, might as well talk about monocytes. Often they say that it's a kidney shape. Well, this is a picture of kidney. I don't really think they look, I guess you have a little tiny curve, so maybe a tiny bit, but whatever. Use your imagination. Monocytes are kidney-shaped nuclei. Complement system. So complements are just these little protein nuclear warheads floating through the blood, but they're technically inactive. But when they get activated, they become the nuclear warheads. They're produced by the liver, and they travel around the blood until they get activated. There's an infection or something like that. So these are a lot of the players, but the most important one by far is C3. So how do these work? Opsonization and enhancing phagocytosis. So the C3b can bind onto a microbe and allow a pathogen, allow a macrophage or something to phagocytize it more easily. Why is this necessary? Well, because pathogens a lot of times are like slippery. They're hard to get a hold of. And so the opsonin, again, we have two. We have C3b, and then as an antibody, IgG also stands for one. It's kind of like a leash. It's a little bit easier to grab onto this slippery frog now that there's a leash. So that's opsonization. That's one way that the complement helps. Another one is that we can cause recruitment and activation of leukocytes. C5a and C3a are going to play a role here. C5a, we've already seen, plays a role in chemotaxis. And here's our taxi. I think of taxi. It's just going to help the neutrophils hone in specifically to the site of infection. And then we can form what's called the MAC complex. Membrane attack complex really means we're forming a little hole, poking a hole in the cell, and then a bunch of water can flood in. And so this osmosis, swelling a cell with water, is just going to make it explode, and that kills it, which is kind of hardcore. So poking a hole in somebody or a cell is not good for it. C3, again, most abundant protein. C3b is the activated form for C3. Again, C3a does some stuff too, but C3b is the main player. So this is the goal of the complement system for the most part. This is going to help kill bacteria. And there's three different pathways to activate C3b. So these are the three pathways. We have the alternative, we have the lectin, and we have the classic. Again, all of these are activating C3b, which can then activate C5, which can activate the MAC. And let's go through these one by one. So again, it helps if you keep in mind that the ultimate goal of every one of these pathways is to activate C3b, which is really just cleaving C3, cleaving the A off. So now it's C3b. And this can ultimately go ahead and activate a C5b, which can make a MAC. So let's look at this. So we have the alternative pathway. So some C3 proteins will just spontaneously, do hydrolysis or cleave off the C3a and just become C3b. But this is very short-lived. It's very unstable unless it's stabilized by a bad guy. So some lipopolysaccharide or fungus or bacterial cell wall viral envelope will stabilize C3b. So C3 randomly turns into C3b, and then it goes away unless there's a bacteria, there's a pathogen there, and then C3b stays around a little bit. It combines with these random proteins, which aren't really that important, and it makes, blah, 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 a C3 convertase. This does exactly what it sounds like. It just converts, it makes more C3b. So it cleaves C3 into C3a and C3b. And so if we think about that, there should be a positive feedback in this. And yeah, absolutely, there's a positive feedback in this. Again, I just have this here because C3b is stabilized by binding to a pathogen. So the lectin pathway. A lectin is just any protein that binds onto a sugar. A lot of bacteria happen to have mannose sugars on their surface. Isn't that convenient? So here we have this bacteria shown here, and we have all of these mannose sugars, and here's a mannose binding lectin. Let's pretend that this is C1. So we'll bind onto this, and this can now activate. C4 turns into C4b. The B is typically the activated one, and then we can also activate C2b. So we have activation of C1. C1 can then activate C4 and C2. So we have these two guys, and these will now team up. So earlier we had a C3 convertase, which came from C3. But you can also make a C3 convertase, which does the exact same thing. Instead of it being made up of C3, it's made up of C4b and C2b. if you remember from this, we saw that these two pathways are a little bit more similar than the alternative pathway. They work both on C4 and C2. So this is the classical pathway, and it's the only of the three pathways that involves the adaptive immune system, that involves antibodies. And a useful way of remembering this is that GM makes classic CARs. So what happens is, again, I have this C1 protein, and the presence of an IgG antibody or an IgM antibody can activate C1. How does it do that? Well, C1 is normally inhibited by a C inhibitor, C1 inhibitor, and this will just basically take off that inhibitor. It takes it off. Now C1 is active. It can do exactly what it just did. It converts C4 into C4b, C2 into C2b, which are active. These things can team up, make a C3 convertase. What does that do? It does exactly what it sounds like. It cleaves C3 into C3b. What does C3b do? Well, that makes C3b turns C5 into C5b. Also cleaves off the C5a, which is helpful for recruitment of neutrophils and chemotaxis. But the C5b then is going to go ahead and help make the MAC complex. What I have IgM highlighted here, it's because so here's our C1 protein. For this to be activated, it needs to bind to 2 antibodies at the same time. And we'll talk about it later, but IgM is a pentamer. It has 5 antibodies at the same time. So because they're right next to each other, it's pretty easy for both of these to bind at the same time, rather than 2 separate IgGs being at the same place at the same time. IgG can activate it, but IgM is just better at activating it. Again, when it binds, this now makes the CI, it swoops off, it leaves, and then C1 activates C2 and C4, and that activates C3b, and that activates C5b. This can do chemotaxis and work with this to activate other immune cells, but we're going to go ahead and now make a MAC complex, which just means that a bunch of water is going to rush in to our cell and cause it to explode. Here they're showing this as a host red blood cell. Well, that's obviously bad. If it's a red blood cell, that would be some sort of like autoimmune thing. But a way to remember this, A for anaphylaxis, asthma, and allergies. This is going to play a role in anaphylaxis, systemic inflammation. C5a in particular is going to be chemotaxis. B for bind. So C3b is going to be our opsonizer, the binder, the handle, the leash of that frog. Again, the other one is IgG, which we'll talk about later. And then I remember that the end goal is like the MAC complex, if you can make it happen. And so it would make sense that this, if we have 9 complement proteins, that it'll be the last couple of complement proteins in terms of the numerics that constitute this. All right, what happens if we are deficient in the complement, and how can this be? How can this happen? So, encapsulated bacteria are particularly hard to grab. They're particularly slippery frogs. So that's why we really need C3b for them. So if you have a complement deficiency, you just no matter what, you're going to be susceptible to encapsulated bacterial infections. Now, more specifically, a C1 to C4 is going to sort of enhance the likelihood of an autoimmune syndrome. So you cannot clear immune complexes. An antibody will bind to a pathogen, and sometimes this just gets stuck in the body. And I think of something like this. It's just it just gets stuck in tissues and your joints especially. It causes arthritis. This is what a type 3 hypersensitivity is. Rheumatoid arthritis and lupus are examples. But let's think about arthritis. These complexes will get stuck in your joints, and then eventually this will cause recruit neutrophils and cause damage, and that's how you get joint pain. Normally, these immune complexes are cleared by macrophages in the spleen. But they can only do that if there's a C3b. Actually, no, I guess C3b would be over here. If a C3b is bound to this, that's the only way that the macrophage can pick this up in the spleen. So without this, the macrophage in the spleen can't clear them, and so they just deposit in the tissues. A C5 to C9, this one's random. Just Neisseria infections are really common with C5 to C9. Okay, so the complement system is really useful, but obviously if it's overactive, it can be bad. So how can we pump the brakes on it? C55, also known as DAF, and MIRL, CD59, these are going to work together. C1 inhibitor, this is going to inhibit C1, and then factor H is going to accelerate the decay of a C3 convertase, whether it's the C2b, C4b, C3 convertase, or the C3b, C3 convertase. All right, what happens if I have a C1 inhibitor deficiency? All right, let's remember exactly what C1 does. C1 is inhibiting the C1 from being active. Active C1 is going to, cause C4b and C2b to be activated. So if I have an inhibitor deficiency, that means this is overactive. The activity of the C1 is overactive. All of my C4 is going to turn into C4b. All of my C2 is going to turn into C2b. For whatever reason, though, the C4 is more important. You're going to have low levels of C4 because they've all been consumed. It's all been converted into the C4b. So the C4 levels are low, and that tells you that you have a C1 inhibitor deficiency. Now, at the same time, C1 has a separate role that's involved in coagulation. C1 can inhibit kallikrein. Kallikrein is going to activate bradykinin. So what does that mean? That C1 inhibitor inhibits bradykinin basically indirectly. So if I have an deficient C1 inhibitor, I'm going to have elevated levels of bradykinin. Elevated levels of bradykinin. What does bradykinin do? Bradykinin causes inflammation. It causes vasodilation. It makes the endothelial cells more permeable, so blood can flow out of the vessels and into the interstitial fluid, into the tissue, causing swelling. And this especially affects kids. And you can see that this girl has what's called hereditary angioedema, or specifically angioedema is just kind of like swelling of the face. And that's specifically because C1 inhibitor causes elevated levels of bradykinin, which causes excessive inflammation. Really high yield to know for this is that if someone has a C1 inhibitor deficiency, you cannot give them ACE inhibitors. Why? Because another thing that can inhibit bradykinin other than C1 inhibitors are the protein ACE, angiotensin converting enzyme. This can inhibit bradykinin, but now if I have an ACE inhibitor that inhibits this, which, an ACE inhibitor inhibits ACE, and ACE inhibits bradykinin, well, that just means I'm going to have even more bradykinin. So however bad this girl's issues are, they're going to be way worse if she also has an ACE inhibitor, because it's double the amount of bradykinin now. Moving on to C55 and DAF, it could also be a CD59 MIRL deficiency. This is caused paroxysmal nocturnal hemoglobinuria. This kind of means intermittent. Nocturnal means at night. So this is going to be basically you're supposed to have these episodes of hemolysis that sometimes get worse at night. People actually debate this. Some people say that this is not true, and so it's a misnomer. But nevertheless, you're going to have widespread hemolysis due to the excessive activation of the complement system. Whenever you break a bunch of blood cells up, this is going to activate a bunch of platelets, which causes thrombosis. So you're going to have clotting issues. Also, you're going to have decreased haptoglobin. Haptoglobin binds free hemoglobin. So if you're doing hemolysis, you're destroying all of these red blood cells. There's a ton of hemoglobin in the blood. It all binds to the haptoglobin and it consumes it. So the levels of haptoglobin go down. Similarly, you have so much hemoglobin in the blood, some of it's going to ultimately get filtered through the kidneys into your urine. So you're going to have dark urine because you have a lot of hemoglobin shown here. All right, inflammation. So it sounds silly, but you just need to know these 5 things of inflammation. And I'm sure anybody who's gotten a boo-boo during a soccer match is well aware of the signs of inflammation. All right, we've talked about this a bit before. Macrophages and phagocytes can recognize pathogen-associated molecular patterns like lipopolysaccharides, like flagellin, et cetera. Like our ribosomes are 80S, their ribosomes are 70S, so that's a pattern in and of itself. But they're also going to go ahead and try to clean up damage. Eventually your cells just get sick and they die, and you need to fix this. So if a bunch of cells die for whatever reason, you're getting old and you need to recycle a bunch of cells, the innards of a cell are going to spill out. And this is a different molecular pattern, a damage-associated molecular pattern, but it works the exact same way, is that you will have your macrophages notice it and then, engulf the dead cell so that it's not taking up space in your tissue. Again, just a friendly reminder that CD14 goes with lipopolysaccharide. All right, so during an inflammatory response, these guys are going to initiate the inflammatory response, releasing a bunch of cytokines, things like histatin, serotonin. We often think of serotonin, SSRIs and happiness and whatnot, but it also causes vasodilation. Eicosanoids, things like the prostaglandins and the leukotrienes that we've seen before, are also going to contribute to vascular permeability. That the blood vessels are going to dilate. Notice we have this room for blood and complement proteins and neutrophils to get out. And yeah. So this is just another fun picture. I think you should really know this. I think it's high yield. All right, we really talked about this to death. Polymorphonuclear leukocyte. What's that? That's just a neutrophil. Lymphocytes can get out. Monocytes can get out. Technically, when they get out, they then are differentiate very quickly into macrophages. Plasma proteins or complement proteins are going to get out. And cool. So neutrophils are the SWAT team. they are short-lived. So macrophages are going to be our cops or patrols, meaning they're present at the beginning of the infection. They're calling for the backup from the SWAT team. But eventually, you win the battle, hopefully, or you're dead. You win the battle, you kill the pathogens, and the neutrophils, the SWAT teams eventually leave, but the macrophages, the cop, they need to stay around for the end. So for anything, for the rest of preclinical, if they're describing like, a heart attack, right? And they're saying, how long ago did this person have a heart attack? If they mention that there's they do a biopsy of the heart, they look at some of the cells and they see a lot of neutrophils. This person had a heart attack like 3 or 4 days ago because they still have neutrophils there. But if I do a biopsy of the heart and I see that there's a bunch of macrophages and T lymphocytes, that's how I know that it must have been a week or more ago. So the neutrophil response is just a week. So macrophages are going to be before the infection, and then like well after the infection, predominantly we're going to have macrophages. Our neutrophils are gone. Obviously, having some short, appropriate timed inflammatory response is good. It's bad. It's pathological. It's bad if you have a chronic inflammation. Contributes to aging. Secondary immune system organ, secondary immune system organ, and then a primary immune system organ. Also, the bone marrow is a primary immune system organ. Let's talk about the lymph node, the spleen, and the thymus. So, you guys do your, this is one of your checks you have to do when you do a physical in ICM. You have to, check somebody's lymph nodes, or if COVID's really bad, then you ask them to feel their own lymph nodes, which is kind of silly. During an infection, these things will get swollen, and that's because this is kind of like the cop station where a bunch of the B cells and the T cells hang out, waiting, waiting to hear who the new bad guy is that they need to come fight. Our whole body is covered in lymph nodes all over the place, and what happens is when blood, is moving through our bloodstream and goes into our capillaries here, we have an exchange. We drop off glucose, we drop off oxygen, and we pick up the garbage. We pick up the CO2, we pick up the urea. And this is, circulatory system stuff that we'll talk about later. An important thing, though, is that you're typically going to push out more fluid than you bring back in. So that means you would accumulate fluid in your tissues over time. This would cause edema. But a bunch of the fluid is just going to be picked up, shown here, into the lymph, and then this is going to travel to the nearest lymph nodes. So if someone has their lymph nodes removed, what do you see in the surrounding area? You see a lot of swelling because they're not good at this lymphatic drainage because they have lymph nodes missing. But a large advantage of this is, imagine you have an infection. What else is getting pushed out of your blood? Not just glucose and oxygen, a bunch of the pathogens are going to get pushed out. And those are going to get picked up in the lymph, and they're going to travel to the lymph nodes. And so this is kind of perfect because the B cells and the T cells are waiting to see who is the invader, or the dendritic cells are going to go, but this is another mechanism by which they're going to go. Lymph nodes. We just said that from the site of infection, sometimes the dendritic cells might come over here and come into the lymph nodes. But we also have antigens that got picked up, from the interstitial fluid when it got pushed out of the capillary. So, maybe this is the protein, this is the arm of a bacteria, and that's the leg of a different bacteria, and maybe this is a little virus. And all of these things are going to go into the one of our mini lymph nodes, whatever the closest lymph node was. So 3 major regions we should be aware of. We have the cortex shown here. We have the paracortex shown here, and then we have the medulla shown there. In the cortex, this is where we're going to have a lot of our B cells, specifically in the follicles. In the follicles, we're going to have B for B cell. What is FDC? This is a follicular dendritic cell. It's just instead of the dendritic cell going from the battlefield to the lymph node, this is just a different type of dendritic cell that ideally is going to pick up this guy and this guy and this guy as they're floating through. It's going to pick them up right here and then turn around and then present them to the B cells on the other side. Also present them to the T cells over here. Which brings me to my next point. In our paracortex is where we have our T cells. In the medulla, we have 2 different regions, really. We have the sinus. So the sinus is right here. And in the sinus is where we have plasma cells, which are just activated B cells, and they're going to basically shed a bunch of antibodies, which will enter the bloodstream. The medulla cords is specifically where the actually, I think I got that backwards. Excuse me. This is the sinus, and these are the cords. So the plasma cells are in the cords, not the sinus. On the other hand, the macrophages are sitting here in the sinuses, and they're just going to filter the lymph. So the lymph is going to come, from the site of infection, or, maybe it's not an infection, just it's going to come from the blood, comes over here, enters, and then it just kind of percolates through this layer. It goes through the cortex, it goes through the paracortex, it then goes through the cords where we have the plasma cells, and then the last stage is the macrophages, which are going to be the final filtering of the lymph, and then they're going to leave. I think it's not super high yield, but B cells and T cells. Remember that they're born in the bone marrow. The B cells go to stay in the bone marrow for differentiation, and then the T cells, go to the thymus. So B for bone marrow, T for thymus to differentiate. These are going to enter into the blood, and we have these things called high endothelial venules, which is basically very, very leaky capillaries. Very much, this process, the rolling and the sticking and the crawling and the transmigration is very similar to what neutrophils were doing, but now it's just the lymphocytes eventually leaving so that the T cells can stay in the paracortex and the B cells can go into the follicles. One thing that's really, really important actually is that the primary follicle is a follicle with B cells that are inactive. During an infection, though, if the right B cell with the right antibody that sticks to the right pathogen is activated, this will mature and turn into a secondary follicle. We have a bunch of activated B cells. Secondary follicles have germinal centers, which are white. I think in my test last year we had some sort of germinal center. So I don't know, maybe, and I think I got it wrong. So just don't get burned by that. Be able to recognize what our lymph nodes look like, and that's a germinal center. That would be an activated follicle, secondary follicle. Okay, what else? The paracortex specifically is when you have DeGeorge syndrome, you're going to have like a kind of an absent dysfunctional paracortex, which we'll talk about later. And if you have an infection, you have a swollen lymph node. I mean, I guess the whole lymph node is swelling, but it's especially going to be the paracortex where a majority of the swelling and the proliferation of cells is going to happen. All right, we really just talked about this all already. All right, the spleen. So the spleen's job, again, we mentioned it, removes encapsulated bacteria, which are very sticky. It needs specifically C3b, and it also gets rid of old red blood cells. Red blood cells live for about 120 days, and then they get recycled by the spleen. It also happens to be a reservoir for platelets and red blood cells. So when the stuff is moving through, so our blood is moving through, it goes through the what's called the white pulp first, and then it goes through the red pulp second. So the white pulp includes the follicles, the periarteriolar lymphoid sheath, and the marginal zone, which respectively have B cells, T cells, and macrophages. I think of M marginal, M for macrophage. This is what's going to be capturing the encapsulated bacteria. So I'm moving through here, and we have our marginal zone, which is on the outside. And it's not really shown in this picture, but, some of this is going to go by the marginal zone. So it'll be in contact with that. A little bit closer in, we have the pals, and this is where I'm going to have my T cells. And then it's also going to sort of go by these follicles. So follicles are going to be the same, it's at least the same name, same general function in the lymph versus the spleen. This is where the B cells are hanging out. The red pulp. So after this, we're going to go through the red pulp. So we're going to go through here, and this is where we have sinusoids, which is where the actual filtering, the recycling, the eliminating of old red blood cells happen. This is also where we store a lot of the platelets. It's just another picture. So the blood's entering here, and we know that we sit on the periphery over here. You have your marginal zone. So here again, marginal zone. And some of the blood is also going to go by the pals. So here's my pals, which has the T cells. So if I'm blood and I'm flowing through the spleen right here, here's a macrophage. Here's another macrophage. So the macrophages are going to grab the opsonized, the C3b bound, difficult to grab capsular bacteria, and they're going to eliminate them. And then here we have our follicles. And eventually we're going to be done with the white pulp region, and now we enter the second part, which is the sinusoids, and this is where we're going to get rid of the old red blood cells. So let's look at better look at this. So here's our follicle, here's our pals, and not shown is going to be the marginal zone on the periphery around this. But after we get through this, now we're going to go through the sinusoid. Notice all these little macrophages hanging out on the outside of these. So this whole thing is going to be a sinusoid. And what a sinusoid, and then we're going to go to the veins, and then we're going to go out of the spleen. So let's take a bigger picture of this. So some of these red blood cells are in open circulation technically before they get into the sinusoid. The sinusoids are purposely built so that they have these, they're leaky, cells can squeeze through them, specifically red blood cells, but they're not that big. You really got to squeeze to get through, to get on the inside. And the idea is that young red blood cells have no problem. They're flexible, they're pliable, they can squeeze right in there, it doesn't damage them. Old blood cells will get damaged trying to do this. And we have a bunch of macrophages, as shown here, here, and here, are macrophages on the outside. Let's say this guy's old and he's having trouble getting through. We're going to come in and we're going to kill this guy. And this is how we get rid of old red blood cells. I thought of it as like, trying to eliminate people by seeing who can do the limbo. And young kids could do the limbo, but an old person would probably have, difficulty doing the limbo. And then we kind of have a midsommar thing on our hands, and we're going to, sadly get rid of the old person. So this is a sinusoid, this whole thing. Often they like to say that they're wrapped in these reticular fibers shown here, and they say that these are straps like a wooden barrel. So that could come up in a vignette. The splenic cords, the cords of Bilirath, are really just those reticular fibers I just mentioned, plus the macrophages on the outside. So. It's not a super important distinction, but it's one that bugged me. This is just a simpler picture. So these things are trying to get into the sinusoid vessel, and if they have difficulty squeezing through, our macrophages are going to kill them. All right, again, capsulated bacteria are slippery. They need to be dealt with by macrophages in the spleen via an opsonization with C3b. And we see that here. Here's my C3b. Here's B for bacteria. Notice there are antibodies, there are complement proteins, and here's my I guess technically this would be a neutrophil, but imagine that this is like a macrophage, which would make more sense for the spleen. It's going to be in the marginal zone, and as the blood flows through here, it is going to be captured. These are going to be flowing through the blood, and they get captured and destroyed by the spleen. So if you have a splenectomy, you're going to have a lot of capsular bacterial infections. And similarly, if you have a splenectomy, you can't get rid of your dysfunctional old red blood cells. So you have what's called Howell-Jolly bodies. This is just the nuclear remnant of a red blood cell. Should not have a nucleus. Usually they're cleared, meaning the spleen gets rid of them, but not if the spleen's either taken out or it's defective for some reason. A target cell is just, you can have issues with hemoglobin, like a thalassemia, or you can have a liver issue, but there's a surface area to volume ratio that's screwed up with the red blood cell. Any defective red blood cell should get cleared by the spleen. So if you just see a shitty red blood cell in general, you probably think that the spleen might be bad. Also, you might have thrombocytosis because the liver is supposed to clear platelets. If you don't have a spleen, obviously, that's going to be an issue there. All right, so B cells, everything comes from the bone marrow, but B cells also mature in the bone marrow. T cells, T for thymus. Here's our thymus, kind of right in the middle of our chest. And the actual T cell development, I think of it as like some sort of intense boot camp. Let's take a look at this. So they start in the bone marrow, and then they go to the cortex. Cortex almost always means outside. Medulla almost always means inside. So we go to the cortex first. So in the cortex, this is where we're going to have positive selection. So all of these, and I think it's a stupid, silly thing, but it's high yield to know when the CD3s, 4s, and 8s are there. So in the cortex, they're in stage 1, it's got positive for everybody. So you have a CD3, 8, and 4 positive. Here, I'm going to see if they can even recognize an MHC. That's the whole job of my T cells. They're supposed to recognize MHC type 2 if they're CD4 and type 1 if they're CD8. If you cannot bind an MHC, then you're useless. So that's where we say positive selection, meaning if you bind, you are selected for it and you get to divide a little bit and go to the next layer. If you weakly bind or you don't bind at all, then you know what, you're useless. We're going to get rid of you. You're dead. So now we're going to the next layer. We're in the medulla. Notice that you've selected a pathway. Your cells are now either a CD8 and a CD4. They no longer have both markers, but they still have the CD3 positive. They maintain the CD3 positive. So we had positive selection in the cortex. It's negative selection here. So here we just wanted to make sure, hey, can you at least do your job? Can you bind an MHC? Here I'm going to make sure, hey, you're not going to go like a little nutso on me and start attacking everything, even our own body tissues. So here I just imagine a scenario where the cells are constantly kind of like, on a silver platter, like, would you like to try some collagen? Can I interest you in some polymerase? Can I interest you in some elastin? It's just going to display all of these normal human self-antigens to the T cells, and they must not recognize them. If they do recognize them, if they bind to the antigen, antigen, we usually think of antigen as a bad guy, but here antigen just means your own normal healthy tissue. In that case, we're going to do apoptosis because I don't want that cell. That cell is going to be a sure recipe for an autoimmune disease if I allow that cell, past this thing. So that's why I think of this as like some sort of, intense boot camp, because there's like a 90% attrition rate, only like 10% the best of the best make it through. And then we get our helper cells and our CD cells, and the helper cells can, further differentiate. All right, intermission. I have another video. I'll see you guys soon. Bye.